Okay. Okay, for others who still not change your ID name, please do so. All right, so we will start today with the new topic, with the new topic of protein synthesis. So this one, we're going to take a look on the functions, okay, functions of the process of transcription, how the protein is synthesized, starting from the DNA, RNA, and how the proteins is become functional. Okay. So we're going to look at the function of mRNA, tRNA, and RNA in proteins biosynthesis. Okay. So this part is different from what we have discussed before in previous topic, where previously we discussed mainly or specifically on each amino acid, how the amino acid is degraded and also is anabolized, okay, one by one. But then for this one, we are going to focus on the at the genetic level, okay, not genetic level, it's some kind of in the transcription level, starting from how the DNA is transcribed into RNA and so on until we get the functions into the functional proteins. Okay, I'm going to share screen. I'm going to, okay, slideshow. Okay, so learning outcomes for these topics. So hopefully by the end of these topics, okay, of these sessions, you will be able to differentiate the functions and structure of mRNA, tRNA, and RNA. So we have a different RNA which play a role to actually brings or translate whatever the genetic the DNA sequence until RNA, and then we get the amino acids. And from there, this amino acid is going to be a 3D functional protein. Okay, and then you are able to describe the function of mRNA, tRNA, RNA in protein biosynthesis, how each of these play their role to the how they synthesize the protein, and then you'll be able to explain each stage of protein biosynthesis, what are the stages that involve, okay, and then we'll be able to relate every functions of cofactors during the protein biosynthesis. All right, and this is the outline or contents of this topic. So we are going to look at the so there are three parts. Okay, three parts involved. So the first part is the mechanism of protein synthesis, which involve the initiations, elongation, and termination. So these are the three earlier steps in the protein synthesis. Okay, and then we will see on the post-translation process and then the targeting process. All right, so for the overview, before we get started to discuss on the specifically on how protein is synthesized. So let's take a look at this overview of protein synthesis. And we know that proteins actually consist of this long sequence of amino acids, right? So this amino acid sequence is actually constructed through the translation of information which is encoded in the mRNA. So how does this amino acid sequence know to attach to which amino acid sequence? So how let's say we have 5 amino acids in one peptide. So how does this amino acid know to to attach to which amino acids, and what's next and what's next. So this is actually constructed through the translations of information from the informations that we get from DNA, which is encoded and then encoded into mRNA. So this is how the amino acids follow the sequence. Okay, and we have actually more than 70 different ribosomal proteins, and this ribosomal protein is going to aid the process of protein synthesis. Yes, we have more than 70 different ribosomal proteins, and we have 20 or more enzymes to activate the amino acid proteases. So during the process of protein biosynthesis, so there are there are actually 20 or more enzymes, which helps to activate the amino acid precursors. Okay, you can imagine we have 20 amino acids, so perhaps 20 different enzymes to actually specialize for each amino acids. Okay, so we're going to look at later which enzymes actually helps to control the activation of each amino acids. And there is also what we call as auxiliary enzymes or protein factors, which helps in initiations, elongation, and termination of polypeptides. And there are 40 or more kinds of transfer in ribosomal RNA. And this is the ribosomal RNA. The shape is like this. And a polypeptide of 100 residues. So let's say we have a polypeptide which contains 100 amino acid residues. These amino acid residues actually can be synthesized in the cells in about just 5 seconds. So you can imagine if just 100 amino acids, so our body can do it, our cells can do it, can synthesize it in just 5 seconds. So the more or the larger the amino acids, or the longer the peptide is, perhaps it needs longer times. But it's very amazing, because it just needs 5 seconds to do this synthesis just for 100 amino acid residues, and then we have about 300 different macromolecules cooperate to synthesize polypeptides overall. So to make it this long polypeptides, okay, we need to involve the collaborations of 300 different macromolecules to actually make this process of protein synthesis completed. All right, so these are the list of amino acids. Okay, this is a common amino acid, which is a 20 amino acids that we have discussed before. Okay, so how each of this is translated by the functions of RNA to RNA and so on. So we're going to look at later how each of these, how they are bind to each other to make up a long polypeptides. Okay. All right. So this story or these topics actually started when this scientist, what we call as Paul Zemanick, in 1950s. Okay. So the first research to actually to understand how the protein is synthesized actually comes when the scientist Paul, he radio labeled the proteins, and how he found out that these ribonucleoproteins particles, is in red like this, which is radio labeled. So he wanted to know what is actually these particles in red, which is the radio labeled proteins or radio labeled patches in the cells. So he found out that this is actually the ribosomes, the ribosomes, which is play a role to actually synthesize the protein. Okay, so this is the history how the process, or his research actually come into this paper. Okay, this paper or journal on during his effort to understand the mechanism of photosynthesis and how the and what types of method that actually he used to understand the mechanism of photosynthesis. And another finding is that actually comes from this two scientists, okay, from Mahon and Semenik as well. So how he found out about these activated amino acids actually attached to this tRNA, forming the amino acid tRNA, and from there that they know that there is actually an enzyme that involved during the synthesis. Okay, so there is another finding from them. Okay, and the third one is the finding from Francis Crick, which is the where he found out about the small nucleic acids, because actually during these times, this Francis, okay, this guy, he never knew that this is actually a type of RNA, but he knew that this is nucleic acid. Is actually serve a role as an adapter to attach to these amino acids and also to this mRNA. So that's all what he knows, but during this time, he still don't figure out about this. This is actually RNA. Okay, so this is the process of protein biosynthesis. So we started with how this activation of amino acids goes, and then we get into these initiations, how the initiations of biosynthesis, and then we get into the elongations. And then it comes to termination and release, in how the after we get a long peptide is formed, and how this long peptide is terminated, how the process of biosynthesis is terminated finally, and we get how this long peptide is released. Yeah, and we go into this folding and post-translational processing. All right. So these are the list of the components that are required for the 5 major stages in protein synthesis. So we have the first part is the activation of amino acids. Okay. So this is the stage of activation of amino acid, whereby we have these essential components like amino acids. Okay. So 20 amino acid tRNA synthetase. We have 32 or more tRNAs, and then we have ATP and magnesium. And for the initiations, we have all these components which helps to initiate the process of biosynthesis. And as you can see here, so each of the process, they have a different components involved, but still each of these components, each of these stages, they have the presence of the form of the energy, which is the, let's say for the first one, we have the presence of the ATP, and magnesium is a form of cofactor, okay, cofactor here. And we have the GTP in the initiation process, and we have GTP in the elongation process. So they have a little bit different types of form of the energy that involved, but still until elongations, they have the presence of the magnesium, which is play a role as the cofactor for the to activate perhaps the each of this process. And all right, so what we're going to look at is the how each of these amino acids. So for, let's say, for example, we have here lysines, aspartate, not aspartate, this is glutamate, okay, glutamate, proline, tyrosines, leucine, histidine, valine, and methionines, how each of these amino acid is. So we're going to look at how each of these actually translated into these amino acids and how they are attached to each other until we get a long peptides. Yeah, so this is what we call as protein synthesis, which involve the initiations, elongation, and terminations. And in order to do that, okay, to study the protein synthesis, we have to involve this RNA catalyzed, okay, because we need to start it from the beginning, which is the DNA, DNA, okay, the DNA sequence, and then we get the we need to input the mRNA to translate the code that we get from the DNA, and then we get the long sequence of RNA, and how after that they are going to be translated into a long amino acids. All right. So let's look at the key components first in protein biosynthesis. Okay, the key components. So the main key components in protein biosynthesis, which is the ribosomes, okay, the ribosomes, and then tRNA and mRNA. So these are the three main components that involve in protein biosynthesis. Yeah, so we have different types of RNA. So and let's look at the differences between these RNA. Okay, RNA. So the first one is the mRNA, or we call it messenger RNA. So what is the mRNA? So mRNA is the linear structure of uracil base. It has it's actually a linear structure with the uracil base. So so in the stage of RNA, it's actually different from the ones that we get in a DNA, DNA, because DNA, the uracil is replaced with a different sequence, and it is a types of secondary structure. We can't actually found this mRNA, which is in a secondary structure, which is in the stem loops or hairpin. So this is a types of secondary structure of mRNA, and so you can see here, this is the DNA sequence, which is the nucleotides of the DNA sequence, where we have the components, the sequence of ATCG, okay, ATCG, on how each of this is going to this ATCG is in DNA, right? In DNA. And how each of these is going to translate into this sequence of mRNA. Okay, so in mRNA, it contains the uracil, which replace the. Okay, what is so in DNA we have ATCG. What about so the uracil in DNA, which actually this uracil replaced in from the DNA? Does the uracil replace adenine or thymine or cytosine or guanine from the DNA? Okay, it replaces T. Yeah. So in DNA we have thymine. Okay, but in the mRNA sequence we have the uracil. Okay, all right. All right, so this is how the secondary structure of mRNA looks like. Okay, so we have the form of a stem loop and also a hairpin. So this is the hairpin lah. Okay, macam ni. Okay, so we have the hairpin. Okay, and we have stem loop, and so it is a form of secondary structure on how mRNA looks like. Okay, so we have hairpin and stem loop. Okay, so in the next one we have the tRNA. So how does tRNA looks like? So it looks like a more like clover leaf structure like this. Okay, okay, this is a clover leaf structure. So this is the how tRNA looks like, and the function of tRNA is it will synthesize protein from the mRNA molecules. So we have here the mRNA, right? The long sequence of mRNA, which is translated from the DNA. And then this tRNA here, okay, tRNA will be will actually carry the amino acids, which is translated by this sequence here over here. Okay, so we have, let's say we have this CUA in mRNA. So the tRNA that will comes in will bring this GAU. They will bring this GAU, which is encoded for the leucine. So this is the function of the tRNA. It will carry in the amino acids following the codes that's already encoded by the mRNA. And so this is how this tRNA looks like. So it's it makes sense if we call it T, because it's looks like the letter T, right? So but then if you look at here in details, okay, more focus on each of the structure of the RNA, how each of these they have a different it contains 2 or 3 amino acid residues at different positions like this, okay, which is specialized or conserved for each positions, right? And so this is amino acid arms like this. Okay, so this amino acid arms is actually encoded for different amino acids. Okay, and we have the 3 prime over here. Okay, and it goes towards this way, and we have another 3 primes anticodon and 5 prime anticodon over here at this loop, and it goes toward this one, and we finally end up with the 5 prime over here. Okay, so this is the structure, how the tRNA looks like. And this positions over here, we have the wobble position, what we call as wobble positions. So what is wobble positions? Okay, what is wobble position? So this is what you have to find out. Okay, what is the wobble positions? And the next one is the ribosomes. Yeah, ribosomes RNA. Here are RNA, and this is much more complex secondary structure. It's very complex with several folds and loops. You can see here we have the combinations of loops in each corner, and how each of these components fold together, okay, forming like a piping or something. Yeah, so we have this to make up the structure is more complex, and this is the examples of the 16S RNA. Okay, and we have over here the 5S RNA, and this 16S RNA is the presence of the numbers of RNAs actually depends on the organisms or species. Okay, so example, we have perhaps for the bacteria, we have a different types of RNA. If the fungi, if let's say mammals, plants, they have a different types of the RNA. So that's why when we want to actually identify different species of, let's say, bacteria or plants or whatever organisms, we are going to extract the RNA, okay, the RNA of those microorganisms, and we're going to see what is the sequence, okay, concept sequence of 16S of these microorganisms or any of the organisms to see what types of species they are specifically. Yeah. And okay, so here is, so just now we look at the how each of the components, how they actually looks like in terms of the structure, and now we're going to look at these functions of them. Okay, the function of them in the process of protein synthesis itself. And we have the first one is the mRNA. So mRNA is actually, they will act as a messenger of the DNA. So we have over here the um, um, we have the case we have so, okay, so we have the DNA, okay, DNA in the nucleus. So this has happened in the nucleus. So we have the nucleus, okay, nucleus. So in a cell, so we have the nucleus. So we know that nucleus is very important organelles in a cell because it contains the fingerprints, fingerprints of each of the genetic, okay, of the organism. So let's say we have the DNA, okay, we have the chromosomes, and we know that in the chromosomes, right? Okay, in each of these chromosomes, we have the sequence of whatever they bring in the code of the DNA of the okay of this. And what happened is that in the first step, this DNA in the nucleus, they're going to be synthesized. Okay, synthesized of the mRNA in the nucleus. So this long chain of the DNA over here is going to be synthesized by this into this mRNA, okay, long chain of the mRNA, and this mRNA will be released through this pocket, okay, through this hole, okay, through this hole in the nucleus wall. So it's going to be released over here as the long mRNA. And then what happened is that the ribosomes, so it will come here in the cytoplasm. So what happened is the ribosomes will come in to actually read through the sequence, okay, read through the sequence that's contained in the mRNA, okay, that contained in the mRNA, and it will be translated into these amino acids. Okay, so these ribosomes over here, we have the large subunit of the ribosomes and small subunit of the ribosomes. So what happened in the large subunit, okay, what happened in the small subunits, and how these ribosomes um, uh, functions to actually translate the sequence in the mRNA to this amino acid. So we are going to look at later in details. Yeah. So this is just overview on how the steps takes place, starting from the DNA onto mRNA, and then the mRNA leave the nucleus through this pore, and then the ribosomes will read through the code that's already encoded by the mRNA from the DNA. Okay, and so that you know the process involve the first one in the nucleus itself, and then the second part is in the cytoplasms. Yeah, and so this, as I said, the tRNA over here will carry the amino acids during the protein synthesis. So we have the function of the tRNA as well over here, will brings into towards this way, okay, to the formations of these long amino acids. But we are not, but still, I'm not describing details. But we're going to look at later how the tRNA comes into the process of protein synthesis. Okay. All right. So this is how the ribosomes itself. Okay, how does it looks like, the large and small subunit. So let's say here we have, for example, we have 50S, which is a large subunit of the ribosomes, and then we have the 30S, which is small subunits of the ribosomes. And this is the tRNA comes in here, tRNA, right? tRNA, which work together with the ribosomes. Okay, and this is the mRNA, and how this tRNA works together with the ribosomes and process the long polypeptides of the amino acids. Okay, so this okay, let's look at more in details on how the functions of the mRNA, tRNA, and RNA. Okay, so we have these, as I mentioned before, we have the DNA in the nucleus. Okay, in the nucleus, and then we have the how the transcription takes place, which code, okay, which translate the sequence of the DNA into this mRNA sequence, the mRNA sequence, and then this mRNA will be split up into this single mRNA, which becomes a major mRNA, and it will go through into this pore and towards these cytoplasms where the protein synthesis or translations will take place by the ribosomes and the tRNA. Okay. All right. So one thing before we go through on the translation process that involve the ribosomes and tRNA, the first thing we need to know is about the genetic code. Okay, the genetic code. So the genetic code that actually that we have in the DNA sequence consists of the ATCG, as I've mentioned. So and there is actually a sequence. Okay, a sequence in the when it comes to the sequence in the mRNA, we have the what we call as triplets of nucleotides, yeah, which codes for specific amino acid. For example, a codon like this, okay, we call these a codons, a triplet of the nucleotides. Okay, so a single letter here is the we call it a nucleotides, and when it comes to 3 nucleotides, we call it as codons, and each of these codons they actually codes for specific amino acids. Yeah? And what are 3-letter code for each amino acids? Okay, so these are the dictionary, what we call dictionary of amino acid codes words in mRNAs. Yeah, in mRNA. So if let's say in a long sequence of mRNA is coded for this UUU like this. Okay, so this is code for phenylalanine. Yeah, phenylalanine UUC is also code for phenylalanine. So different amino acids, they have a different codes, and each of them amino acids they have perhaps more than one codon, which is code for each of the amino acids. Because if you look at here, we have serine. Serine have four codons, which codes for serine. So we have UCU, UCC, UCA, UCG. So this actually just specialized for serine. Yeah? And okay, so so do you understand how to see this dictionary? Okay, so we started with the U here, and then U. So let's say first letter code is equal to U, and second letter code is U over here. It represents YU. So we have UU, and then the third letter. Okay, the third letter is different from for each amino acids. Okay, so we have, even though we have UU over here, but it just not precisely code for just not for phenylalanine, but it's still, but the third letter code will mean the precisely on what are the amino acids that they are coding for. Okay. And if you look at here, leucine is actually have more than 2. They have 1, 2, 3, 4, 5, 6 codon that represents themselves. Okay, represents the leucines itself. And then yeah, we have alanine. We have 4 codes. Threonine as well. We have 4 codes for codon. So different of each of them, they have a different one. Okay. And Okay, so this is another way how to see the how this each of the amino acids is coded based on the codon. Okay, so if we have here is in the round shape like this, okay, like wheels. Okay, so we have the UCAG like this, and the second layer is another um, which is represents the second letter, okay, second letter code, and then the third layer is the represents the third letter code. So we have UUC, which is represents for phenylalanine, UUU represents for phenylalanine, and yeah, and so on for another amino acids. And apart from the codon that we have discussed, yeah, the three letter codes that's coding for each amino acids, we also have the anticodon. Okay, anticodon. The codon is, we know that it is actually the three nucleotides which code for the proteins or the amino acids. And the codons at a three-base sequence on the RNA, we call it anticodon. So we have here the codon which is on the mRNA, okay, on the mRNA sequence. So we have the codon which represents each of these amino acids. But then when we want to translate this codon in the mRNA, then the at the first place, this codon will be translated into this anticodon, which will brings in by the tRNA. Okay, so the A will be translated into U, into A here over here, and into G over here by the tRNA, which is what we call this as anticodon. Okay, what we call is anticodon. Then this anticodon actually will be translated later on by the what are the amino acids by the tRNA. And okay, so this is the wobble. So wobble codon or wobble base is actually or the letter the third letter, okay, the third letter, what we call as the wobble base of the codons, actually contributes to the specificity, of what types of the amino acids actually will be translated by the tRNA. Because okay, the wobble, okay, the wobble base or wobble, the third letter is actually have a very unique characteristic where it's actually the third letter, okay, it will pass loosely to its corresponding base in the anticodon because it will permit the rapid dissociations of the tRNA from codons during the protein synthesis. Okay, so this is a very unique characteristic of the third base, okay, a third base, because we know that once the tRNA bind to this mRNA, okay, with this which bring this anticodon, then the third letter, they actually will just loosely bind to the third one, okay, to third codon to allow the releasing, okay, to allow the faster or fast releasing of this anticodon from this mRNA. So to allow for another tRNA to come in. Okay, so that is the specific characteristic or a unique characteristic of the third base in the codon. All right, and okay, I think that's all the class for today because I'm not going to go into details into I'm not going to start with the stage one. We're going to start this one on the next week on Monday. So today I just covered on the on the RNA, what are the important components that will aid in the protein synthesis. So we have discussed on the different parts of different functions, different structure of the mRNA, of the RNA, which is mRNA, gRNA, and RNA. So these are very important components that you have to understand the structure, you have to understand the functions, each of them, so that when we apply these three components into the process synthesis, it's very helpful for you to understand the process. And yeah, I think that's all for today, for today's class. Okay, for today's class. So we are going to start with the stage 1, which is activation of amino acids, and until the end of this topic lah, hopefully by this Monday. And I think I'm going to do the class on Monday face to face. So we hopefully we can start face to face again before one class or few classes before the semester break. And so this is the so today you're going to have the task on. So hopefully everybody can come to the makmal pengajaran. Okay, they want to, bukan they want lah, okay, the makmal on time. Okay, so hopefully by 2:30 everybody is ready, just to, you know, the paper, the exam paper is already there in front of you, and when 2:30 you can start answering your paper. Okay, so that's all, I guess, for today. So any questions before I end the session? If no, then so good luck for today. Okay, good luck for today's test 1, everybody.